Hi everyone, welcome to today's virtual demo. We're so glad you could join us. I'm Lisa and I will be your guide through today's adventure in the lab. Before we dive in, just a few quick housekeeping notes. Today's presentation and demo will run about 40 minutes, followed by a live Q&A. To keep things running smoothly, we've pre-recorded some sections of today's demo. If you have any questions, drop them in the Q&A panel at any time, and we'll tackle as many as we can after the presentation. And yes, we're recording today's session and you'll get a link to it in a couple of days. All right, let's get to the good stuff. It's my absolute pleasure to introduce our speaker, Dr. Jessica Hartman, Senior Director of Applications and Lab Services at Cell Microsystems. Dr. Hartman has an incredible background in biology and molecular cancer biology, with a bachelor's degree from the University of Virginia, a PhD from Duke, and postdoc training at both Baylor College of Medicine and Duke. She's led R&D programs across biotech companies and now heads the charge developing next-generation cell biology workflows at Cell Microsystems. So sit back, grab your coffee, and get ready for a great session. Dr. Hartman, the virtual stage is all yours.
Thank you so much for joining us for today's webinar. My name is Jessica Hartman. I'm the Senior Director of Product Applications at Cell Microsystems, and you're in our R&D lab today to perform an experiment using MB49 cells that have been genetically engineered to express either GFP or RFP. Before we get started with the lab portion, I'm going to give you a brief introduction to our consumable and our technology to orient you to what you're doing today, and then we'll get into the lab and get started with our actual in-lab workflow. So before we get started with the hands-on portion of the demo, I wanted to spend a few minutes introducing you to CellRaft Technology and showing you how you can use this technology to increase the efficiency of your cell line development workflows. So if you're joining me today, you are probably already doing this type of work and you are interested in finding ways to increase the efficiency and the throughput of these processes. Now, for most of us, this workflow starts with a population of cells that we are interested in genetically modifying, and this could take the form of a knockout or a knock-in. But overall, the goal is to put this material inside the cell. That can take the method of liposuction, transduction, electroporation, or even microinjection into the cell itself. Once you have transduced your cells, you need to turn them into a cell suspension that can be individually interrogated to determine whether or not you've achieved your gene edit. You may have done this using one of these methods, for example, laser-based high-speed flow cytometry, manual limiting dilution with a multi-well channel pipetter, or one of the newer low-pressure droplet dispensers. But regardless, the goal is to put a single cell in a well and then use that single cell to identify your gene edit and hope that that single cell can recapitulate the cell population of interest that is now genetically modified. Despite all of the tools that are available to us in the lab, after we did a poll asking researchers how they saw gene editing workflows in their own lab, it turns out that almost everyone thinks this process is incredibly inefficient and very time-consuming. These inefficiencies and time-consuming issues lead to a lot of other consequences, such as a lack of clonality, too few clones and too many consumables, as well as lengthy multi-instrument workflows. So why is gene editing so challenging for most of us? That's largely because each of these steps along the process has its own issues that's going to make it difficult to achieve your end goal of a stably edited cell population. The very act of transducing your cells can cause cell stress that make this process challenging. These processes are typically inefficient, they can cause cytotoxicity to the population, and this often leads to heterogeneity in that population where you may have some cells that are not edited at all, some that only have one copy edited, or in rare cases, only a few cells that have achieved the edit that you're interested in. Once you turn that cell suspension into a suspension and goes into one of these technologies, you're creating additional stresses on an already fragile population. Using fluidic-based manipulation or manual pipetting can cause fluidic pressure as well as cellular damage. And ultimately, the fact that you've manually pipetted or even droplet dispensed a single cell into the well means that that stressed out cell is now sitting alone in that well with no cellular crosstalk from other cells in the dish. And 90% of the time, what this leads to is a single dead cell in a well that is not going to give you your gene-edited population of interest. So really, if the key features here are that we have this inefficient process that's causing damage to our cells and we're left with a cell alone in a well, wouldn't it be nice if we could do this in a flask-like culture environment that also allows us to enable spatial segregation? And that is where CellRaft technology comes in as a totally new way of thinking about cell line development. Our process starts with our CellRaft array, which is shown in cartoon here. It allows us to seed thousands to tens of thousands of cells in a single small footprint, while also enabling us to perform spatial segregation in the individual microwells that we fabricate inside the consumable. When we're ready to use the dish, we take that prepared cell suspension after transduction, and we can simply manually dispense it into the dish, which is a very quick process, and I'm going to show you exactly how we do this when we go back into the lab. Once you've manually dispensed the cells into that footprint, the cells will settle down into the individual microwells within the array, down onto what we call a cell raft. The cell raft is a releasable polystyrene growth surface. So as the cells fall into the microwells, they will attach to that polystyrene growth surface just like they would in any other tissue culture consumable. We can put that dish back into the incubator and we can then monitor growth of those cells over time. So while there may be empty wells or rafts that have more than one cell, we really only care about those clonal rafts, and we can find those clonal rafts and monitor clonal growth throughout the duration of the experiment. Once the cells have grown to a sufficient size that we want to retrieve them, that cell raft is fully releasable from the array and we can retrieve it for downstream recovery and further expansion. And I'm going to show you how that works both in this presentation and then once we get into the lab. Now, I showed you a cartoon of one raft that we were interested in, but the amazing thing about the CellRaft array is that this is occurring on a basically unprecedented scale. That one small footprint contains 40,000 individual positions that we can look at to identify clones of interest. And so in this particular example, you're looking at a stitched together image of every single raft in every single field of view in that footprint. And each of those red colonies represents a pluripotent iPSC clone that we could potentially recover for downstream isolation. That means in one small CellRaft array, we are replacing dozens of 96-well plates and the accompanying media, etc., that is necessary to support the expansion of single cells in 96-well plate consumables. Now, while thousands and thousands of rafts sounds amazing, if you're anything like me, the idea of looking through each of those rafts individually does not sound appealing, especially if you're on a deadline or you have deliverables to achieve. That is where our hardware and our software come in to help you in this process. Our software, CellRaft Cytometry, allows you to perform image-based software-guided cell raft selection. What that means is that we can use the images that we acquire on our instrument to look at every single raft in the array and interrogate it for our phenotype of choice. That could take the form of, I want to find every single raft that contains a single cell at time point 1, and I want to define what a cell looks like to me using size and shape morphometric characteristics. We can also monitor the growth of those cells over time using confluence-based metrics. And if we're fortunate to have a fluorescent marker, we can also use fluorescence to identify our clones of interest. Once we have used the software to identify our cell rafts of interest that contain our clones, we're able to very easily map those cell rafts for isolation. So when I say isolation, what do I mean? We call this intelligent isolation of the cell rafts using our instrument, the CellRaft Air. The Air is shown here on the left-hand side of the screen. You're going to see it in the lab in a few moments, but there are some key features that I want to highlight about the instrument. What you see here is pretty much what you get. It's a very straightforward instrument. There's no fluidics. There's no plastic tubing, no sheath fluid, absolutely no pressure to disturb your cells. It's a very gentle, fully automated system. And the key feature here that I want you to remember is that we're going to isolate the cell rafts themselves. We're not isolating single cells or the colonies. That means that there's no trypsinization, no scraping, no aspirating, no pruning or dissecting of your cells. They're going to stay attached to those cell rafts through the entire duration of the experiment, and we're going to transfer the cell rafts containing your clones at the end of the experiment. We're going to fill a 96-well plate with already established colonies that meet your specific criteria, not single cells, which means we're going to be far more effective than any other technology that puts a single cell in a well. The other reason this is so effective is because this is completely manipulation free. The CellRaft transfer happens without you having to disturb your precious cells, and the cells grow without any further perturbations on your end. So to show you what this looks like in cartoon, once we have identified a raft containing a clone of interest, we use the air instrument to dislodge that raft from the array itself, and it can get transferred in an automated fashion using the air system over to that 96-well collection plate. The collection plate is filled with fluid of choice. That's your growth media that is appropriate for your cell type. And as the raft falls down to the bottom of the microwell plate, your cells will passively grow off the raft attached to the floor of the 96-well plate and grow like they would in any other normal tissue culture dish. To show you what this looks like in real time, this is a bright field movie taken from a transferred cell raft. You can see over about 10 days, the cells that we have transferred on that cell raft that you see here in the middle are growing off the raft and colonizing the floor of the 96-well plate. The raft will eventually dislodge during regular culture and be washed away through that process. Once the colony has achieved a substantial size, it can be passaged through normal methodologies just like you would in any other normal workflow in your lab. Now, the great thing about the technology is that we're transferring these fully formed colonies, and they're going to get you started with your experiment much faster. So what you're looking at here is an example of a CRISPR editing workflow in A549 cells that was performed here at Cell Microsystems. And you can see in the top image, after only 11 days post-transduction, we have a beautiful homogeneous clone that has grown off of that cell raft that you see here. And it was ready to split, and you can see lovely viable cells only 24 hours post-split here. In contrast, after 11 days post-transduction and limiting dilution, we only see a handful of small colonies that are very heterogeneous, are likely not clonal, and we're of course not ready for passage and expansion at the 12-day mark. But even more importantly, because of the track and trace imaging that we're able to do with the air system, we aren't just limited to looking at that end clone. We're able to go back and we can look at every single time point that we took on the air system and confirm that we do in fact have a clonal cell. So what you're looking at is that single cell at day 0 on the cell raft that led to that beautiful clone that you see here at day 11. So this gives us a high degree of confidence that the cells that we're transferring are in fact clonal, and that we can use that information to have a high degree of assurance that we are in fact looking at a gene edited clone as opposed to a polyclonal population. In limiting dilution, this would be basically impossible. If you've ever gone to a 96-well plate or a 384-well plate and tried to find a single cell in a well, you know that is basically impossible and likely to make you more frustrated than confident in your clonal population. Now, these are bright field images, but if we are fortunate enough to have a fluorochrome, our life is even better with the air system because we are able to use the technology to stain, image, and isolate, as well as recover characterized cells using fluorescence. For those of us who have done antibody staining or fluorescence-based imaging using a microscope or a high-content imager, while you can generate beautiful images, for the most part, you can't actually recover a single cell that you see based in those images. On the flip side, you can use fluorescence to do flow-based cytometry using a high-speed sorter, and in those methods, you can easily recover your fluorescent cells, but you can't actually see what they look like in situ attached to a surface. With the air system, we get the best of both worlds. What you're looking at here on the left-hand side is a population of suspension cells that were stained with an antibody on the cell surface. And you can see here that we can very easily identify single cells that are positive for our marker, and those cells are in fact dividing on the system and can be recovered for further expansion. On the right-hand side, you're looking at pluripotent stem cell clones. These were stained with TRA-160 for live staining in the red color that you see here on the image. And we can use that red imaging to identify our clones that are still pluripotent, giving us a high degree of success from these fragile and difficult to grow cells off of the air system. So we like to say goodbye to sorting and hello to rafting because it's really the combined power of the technologies that I've introduced to you today that allow us to be so efficient in these cell line development workflows. We use CellRaft Array to generate the flask-like culture conditions as well as performing single cell separation. That gives us a high degree of success of our single cells actually forming a clone we can use. We use the air system to perform that serial imaging to identify the cells that we're interested in. And then we can use our image-based software-guided selection to actually mark those cells for recovery. Using the air, we can then perform automated retrieval of those target colonies, letting us get to a fully functional expanded clone much faster than any other workflow. So now that you have a better understanding of CellRaft technology and how it can help accelerate your cell line development workflows, we're going to head back into the lab so that you can actually see how easy it is to get started with the technology. I'm going to show you how to prepare and seed the CellRaft arrays with your population of cells. We're then going to scan those seeded arrays using our instrument, the CellRaft Air. And then we'll go out of the lab so I can walk you through how we use our software to analyze the rafts and map them for isolation. We'll then isolate our clonal colonies, and I'll show you how we monitor clonal outgrowth. So let's get started. So we are ready to get started in the lab. The first thing that we have to do is to take our T-flask of MB49 cells, trypsinize them into a single cell suspension, and seed them onto our consumable, the CellRaft array. You can see what the array looks like here. It comes bagged like this, and it has that red dot in the bottom corner that indicates that it has been gamma sterilized. The label on the bag tells you what kind of array that you're using, as well as the ordering information. And for today, we're going to be using a 100 single array. So you know it's a single because there's one central reservoir in the array here. And we have 100 micron by 100 micron rafts, which are the size of the rafts that the cells will actually attach to. So we're ready to take this array into the hood and unpackage it and prepare it for cell seeding. So we have our CellRaft in the hood ready for cell seeding. And to get started with the array, we really only need three things besides the array. We need an aliquot of pre-warmed PBS, which has been warmed to 37 degrees Celsius in a water bath for about 30 minutes to an hour. We have a conical aliquot of our media of choice for our cell type. And we have our prepared cell suspension, which is ready for cell seeding. When we're ready to get started with the array, we're going to remove it from its packaging by simply getting rid of that outer bag. You can see that it comes double bagged, and all we have to do is open up that standard packaging to remove the array. You can see the array here. It has a very familiar form factor for most cell biologists. It's a 60 millimeter size cassette with that interior reservoir where we're actually going to seed our cells. So the first thing that we have to do is to apply 3 mils of that pre-warmed PBS onto the inner reservoir of the array. And we do that by simply dispensing the PBS into that inner reservoir and ensuring the entire reservoir is coated. We're going to repeat that twice more for 3 minutes apiece, and then we'll perform a media wash to ensure that the reservoir is fully washed and ready for our cells to attach. So I have a pre-prepared array here that has already had its 3 by 3 PBS washes that is ready for its final media wash. So to do that media wash, I'm just going to aspirate that PBS using a standard vacuum aspirator. And we will add 3 mils of the culture media that is appropriate for the MB49 cells. Like so. Now at this juncture, the array is ready to go for cell seeding. You could put the array back into your tissue culture incubator while you get your cell suspension ready. Or if you already have your cells ready to go at the appropriate concentration like we do, you can go right to cell seeding at this point. To seed your cells onto the array, we're going to aspirate that final media wash. And then we will use a P1000 for increased control in order to seed our cell suspension. So we'll be putting 3 mils of that cell suspension on the array. And all we have to do is ensure that our cells are nice and evenly suspended. And we're going to seed it in what I call a snail pattern in sort of this drop-wise orientation in order to ensure even coverage of the cell suspension onto the array. That's 1. 2.
And just that quickly, we have seeded and individually segregated 40,000 MB49 cells into those individual microwells. So you can imagine how much faster this is than trying to individually limiting dilution seed 40,000 cells into 40 or so 96 well plates using a standard multi-channel pipette. This array is now ready to go into a tissue culture incubator for cell attachment to the rafts, and we'll go ahead and put that into the incubator now. Now that we have our cells seeded, we'll return the array to the incubator to allow the cells to attach before we perform our first scan. Our cells have had time to attach to the array, and now the array is ready for its first scan using the CellRaft Air instrument. I'm going to show you how to turn on the instrument as well as to create a data record for our first scan. So to get started with the instrument, all we have to do is push that silver button on the front to turn the instrument on. The blue lights light up so that you know that it's ready to go, and we're ready to get started with software. We're going to load in our array that we seeded earlier to perform our day 1 scan. To do so, we're simply going to put the array onto the left-hand side of the deck in the adapter plate, ensure that it is firmly seated, and close the door. Now that we have loaded the array, we're ready to launch the software to perform our first array scan. To do that, we're going to type in that white box in the center a unique name for our array. We're going to call it Live Demo, and we'll click the search button. And the software will not be able to find that data record because it doesn't exist, and will ask us if we want to create it, and we do. So we click Create. We have to tell the system what type of array we're using. We seeded a 100 by 100 single array, and then we have to choose where we're going to save the data. You can choose to save it onto the hard drive of the computer, or you can save it onto an external hard drive for offline data analysis later. And then we choose the collection plate that we're going to isolate into. We can now click on that full array scan button and click Continue. And it's going to do a really critical step in the process, which is the calibration. The instrument's going to autofocus on the array that we have loaded and find the top left corner of the array. This step is critical for ensuring that we are able to capture every single field of view every single time. And that also means that we don't have to keep the array on the instrument to perform that time course imaging. Now that the system has calibrated, we can manipulate our fluorescent exposures. You can see here that I'm changing the exposure times on both the green and the red channel while leaving the blue channel off. We can blow up the field of view to make sure that we can visualize the cells easily that we want to capture in fluorescence. And once we're happy with those settings, we can go to the Array Scanning tab and launch our scan. So now you can see in real-time imaging what's happening on the instrument. That green box on the left-hand side is showing you the field of view that's being captured. And we're going to capture every single field of view in the array in all 3 channels at the same time. So that's what ensures our rapid imaging. You can see it's only going to take about 13 minutes in real time to capture the entire array. All of these images are auto-saving right now, as well as being analyzed in the background. So as soon as the scan is complete, the data are ready to be analyzed to determine which rafts contain your cells or colonies of interest. At this point, we could walk away from the system because this is all going to happen fully automated without you paying any attention to it. And the array can be removed once that scan has been finished and completed, and you could go on to scan another array. We recommend that you scan the arrays at least once a day or every doubling time to capture that full growth record of the cells on array. And that time-bound imaging is what's going to allow us to actually determine which of those single cells turn into colonies. So through the magic of time, we have sped this up so that you can see what it looks like after the cells have had an opportunity to grow on the array. You can see now instead of single red and green cells, we see beautiful red and green colonies because those single cells have turned into happy, healthy clones. At this juncture, we're going to take this data record into the office in order to find which of those rafts contain single cells that we can map for isolation. So we get to take off our lab coats for a little bit as we come into the conference room in Cell Microsystems to do our data analysis on the array that we're going to isolate today. The really nice thing about the software is that it's fully offline, allowing you to analyze your scans whenever you want to, as well as to do it offline from the instrument, freeing up the instrument for other users to perform scans or isolations. So what I'm going to show you now is how we turn that list of 40,000 cell rafts into a curated list of rafts containing exactly the cells or colonies that we're interested in. For today's experiment, we are going to look at our scans over the last 4 days, identify single cells at time point 1, find the cells that were red and that turned into red monoclonal colonies, and then we'll map those colonies for downstream isolation. We're going to do all of this using our software, which enables us to set parameters and really clearly define the cells or populations of interest that we're looking for. So let's get started.
So we are now in CellRaft Cytometry. We're going to click on View List and open up the data record that we have been scanning our array into, which is Live Demo. Double click to open it. It's going to load in all of those images and all of that data that we've been acquiring over the past several days. And you can see here that now there's a list of scans from this array that we've been taking over the last several days as the cells have been growing on the CellRaft. We have 3 modules available, but we're going to use CellRaft Cytometry today in order to take that curated list of 40,000 rafts and turn it into a much smaller list of cell rafts containing exactly those red single cells that turn into a red colony that we are interested in. Now, when you come into cytometry, you're faced with an empty screen like this, and you have a couple of choices. If this is a brand new cell type that you've never used before, you can start from a brand new population where you can predefine all of the parameters that are available to you for your particular cell type in order to find those criteria that look the best to you. Alternatively, you can use our import population feature here and go to our library, which gives you a variety of options to get started with as presets that allow you to use the most common features that we recommend for these various settings, such as single cells, dividing cells, colonies, etc. Now, we're lucky today because we are using an optimized cell line that we are very comfortable with, meaning that we have parameters that we know are going to be very effective already for these MB49 cells. So I can go into an old experiment using this cell line and import the populations that I'm interested in order to get started very quickly with the software. So for today, I'm going to import our single cell subpopulation as well as our confluent colony population and import those. When those load in, you will see that each of those populations contains a variety of parameters that we have found useful for defining the physical and measurement features of the MB49 cell lines. Now, of course, because we're looking for single cells in this first population, we want to go look at our time points and make sure that the single cells are actually set to look for the cells at time point 1, as you can see here at scan 1. And we can also manipulate all of these criteria here to ensure that we are finding our cells of interest. So if we click on the little pencil, for example, for AOI area, you can see a histogram of all of our cells, allowing us to define where our single cells are in that particular population. So we can change the parameters if we wish to ensure that we're only capturing those single cells. And we can pull the data, we can manipulate the histogram to our heart's content. Now, of course, I've already preset these, so I'm going to not save any of those settings. And you can change any of the parameters that you actually would like to change for finding the cells of interest. To ensure that we have actually found single cells that we care about, you can click over here at RAF View, and then you can toggle through the RAFs and find examples to make sure that your settings are actually what you are interested in. So if we look at the query view, we've now used that population to turn those 40,000 cell rafts in the array to a population of around 6,000 cell rafts that each contain a single cell. Of course, those cells are not necessarily red cells. We have scanned both red and green cells in this array, and we're only looking for rafts containing single red cells. So to find those red cells, I can take those preset parameters, click on the pencil, and then add in a feature to allow us to look for red fluorescence. So if I click on that red fluorescence button here, I can then add that in as a feature to continue to triage our list down. So we click Save, and now you can see that list contains a filter set here for red fluorescence. If we click on the pencil, we can see the pencil pop up, and there is our red fluorescence intensity histogram. So we are going to only look for rafts containing cells that are greater than, let's say, 1.5 times higher than background. And you can see here an example of what a cell looks like with that level of intensity. So we click Save. And now what you can see is that list of 6,000 cell rafts has turned into a list of 877 cell rafts containing an RFP positive MB49 cell. So if you were doing any other workflow, you'd have to stop here because you wouldn't have any other data to tie to your single cell rafts. But because we've been scanning these rafts over time, we have data throughout all of those scans that can be analyzed to help us look for cell growth of those single red cells as they have expanded on the raft. So for today, we're just going to look for colonies that have become confluent in the last several days. We're going to click on that pencil here, and we're going to change the time point to our last scan, scan 5. We're going to use this particular example here, this metric called raft coverage, to enable us to set thresholds for how confluent we want our raft to be. This is a really nice feature that allows you to help select for rafts that are not polyclonal, but rather monoclonal, for example, because we wouldn't expect a single cell to have created a fully covered raft in only several days. So we're going to set it here to about 50%. And again, you can see that there are thousands and thousands of confluent cell rafts available to us. But of course, we don't want just any confluent cell raft. We want cell rafts that are only RFP positive. So we can select for that by again going into edit our population. We can add in features for red and green fluorescence shown here. And what we're going to do now is say that we don't want any cell rafts containing any green cells whatsoever because we only want clonal cells. So we're going to change that histogram to below. So we only want rafts that have areas of interest that are below a threshold of 1, for example. And we're going to click Save. And we're going to make sure that those rafts are all red. So we're going to click on the red pencil, and we're going to say that we only want rafts that have areas of interest above, let's say, 1.5%, 1.5 times higher than the background. As we can see here, we click Save. We've now turned that list of almost 10,000 rafts into about 860 rafts containing rafts that are red, but not green or blue. As you can see this here. Now, this, of course, does not tell us that these rafts contain clonally derived populations of cells. So this is where the real power of the software comes in, because we can use all of this data to create what we call a population set. If you come over here and you click on this set over here, you can see that we have the option to add in our populations into our population set Venn diagram. If we click on the pencil here, I can put single cells in the left-hand side in bubble A, and I can put the confluent colonies on the right-hand side in bubble B. And if we look at the intersect of these two populations, you can see that there are about 250 cell rafts that started from a single red cell and ended with a red colony. And to prove it to you, you can see that here is our colony at the last time point. You can see that it is indeed red and not green. And if we track back to time point 1, you can see that that time point 1 scan does indeed contain a single red cell that is not GFP positive. So this list now contains 250 cell rafts that have basically red clonal MV49 cells that we would consider to be ready for isolation on the CellRaft Air instrument. And of course, you can check all of these if you wish. And if you don't, they are ready to be mapped for isolation. You see a nice heat map of what it looks like, all of those clones distributed throughout the array. Each yellow dot represents a single cell raft that meets those criteria. And we can very rapidly map those cell rafts for isolation by clicking on the Sample Randomly tab and then clicking into the cartoon of a collection plate here, allowing us to very quickly fill up that collection plate with our curated cell raft list. And you can see that happening now. And in about 2 seconds, I have now mapped this collection plate full of 96 cell rafts that have red monoclonal colonies that are ready for isolation. We can check that to just double check by clicking on an individual well. And you can see indeed we do have a beautiful single red cell here. And we can find a non-GFP beautiful RFP clonal colony here that's ready to go. At this juncture, we can take this data record back to the air instrument, and we're ready to retrieve all of those wonderful colonies.
The cells on this array are ready for isolation. So we have our array here as well as our 96-well collection plate. We have pre-filled this collection plate with 100 microliters of growth media. You can see that we're using a black-walled collection plate for today's experiment because we're doing downstream fluorescent imaging, but you could certainly use any clear-walled tissue culture plate that you might have in your lab. In order to do isolation, we need to ensure that the media volume in the array is 5 mils. As you might remember, we seeded this array in 3 mils of media and cells. So today we're going to just add up 2 mils of media into that central reservoir to ensure that we have sufficient media for isolation. I'm going to do exactly what I've already done, which is take 1 ml of media using a pipetter, and I'm going to gently add it into the corner of the reservoir. So now we've added 1 ml, and then we'll add a second. And we are now ready to take our array and our collection plate over to the instrument to get started with isolation. To get started with isolation, we're going to load our array that is ready to isolate on the left-hand side of the deck, just like we did before. But this time, we're also going to add in our pre-filled collection plate and load it on the right-hand side of the deck, matching A1 to A1. To install the wand for isolation, you simply click it into that wand holder there, and we're ready to go. We'll remove the lids and ensure that the plates are still seated firmly, and shut the door.
We have the software all ready to go to start isolation. You can see in that center heat map, those blue squares represent all of the rafts that we're going to isolate. And all we have to do now is click isolate. So you can see here that it's going to find the first raft for isolation highlighted in yellow. We're going to zoom in so we can see the raft. This is actually a live feed of the software looking at the raft that's being isolated in that red crosshairs. We're now going to do that needle poke through the floor of the array, and just like that, that raft is gone. So once the software realizes that the raft has been isolated, it's being transferred over to the collection plate where that yellow circle is over there on the right-hand side. And the software will now go through the array to identify all the rest of the rafts that have been mapped. And it's going to do this completely hands-free. At this point, we can walk away from the computer, although we'll stand here for a few more minutes to watch a couple of isolations happen. So it's just that fast. We find the next raft with the clone that we've mapped. We're going to actuate through the floor of the array to poke out that raft, and then you'll see it start to dislodge. And then once it's out, the wand will pick it up and transfer it over to the collection plate.
Our isolation is now finished, so we can remove our collection plate and our array. If we're finished with the array, it can go back into the incubator if we want to isolate more rafts later, or it can simply be disposed of like any normal tissue culture dish if you're done for your experiment. We're going to open the door, remove our collection plate and our array, and we can shut the door. Now here's the best part. We're going to take the collection plate and we're going to bring it over here to our microscope and find those cell rafts that we just isolated. And you can see here in the bright field, there is that cell raft with our precious cells that we just transferred safely attached to the raft. And of course, if we scroll through the microwells, you can see that each of them contains our cell raft with its precious cell cargo still attached. And of course, you could look at all of the rafts right now, or you could put the plate back into the incubator to continue culturing. And what I think is so remarkable about this is that it's so much easier to find that cell raft in the microwell compared to a single cell or even a small colony of cells that's been growing for weeks after isolation. And if we zoom in one more magnification, you can see quite nicely that those cells are still attached to the raft. They're happy and flat and ready to continue growing to propagate your colony of interest. Now, of course, we just freshly isolated this plate, so the cells have not yet had a chance to grow off the raft. So we have a plate that's been culturing in the incubator for several days post-isolation to give you a good example of what it looks like when the cells begin to grow off raft and continue to divide up. And there it is. You can see that beautiful colony that is growing off the raft. You can see the raft here in that dark square right there, and the cells beginning to grow off and attach to the floor of the 96 well plate. This is happening completely passively. You don't have to intervene at all. The cells are just going to do this all by themselves. And all you have to do is wait for the colony to be a sufficient size in order to passage it for expansion to a larger collection vessel or for any other of your downstream experiments. And of course, you can see lots of different examples of what these cells look like. And you can have a complete data record of all of your clones as they've grown off raft in that collection plate. So our 96 well collection plate with all of our precious cell rafts in it is done now, and we can put it back in the incubator to let the cells continue to grow. Of course, I demonstrated the MB49s for you today, but we have tested this workflow with a wide variety of cell types, ranging from primary cells to 3D organoids to stem cells. And if you're interested in learning more about the cell types that we've tested, you can head to our website and check out our cell line atlas, which gives you a very comprehensive listing of every cell type that we have validated here at Cell Microsystems, as well as with our customers. So now that we've finished for the day, I'm just going to shut down the instrument. And I want to thank you all for joining us so much. And if you have any questions or you're interested in demoing the instrument in your own lab, again, please visit our website at cellmicrosystems.com and drop us a message.
Thank you so much, Dr. Hartman. That was fascinating. Before we jump into questions, we've got a quick poll to get your feedback. Let me launch that real quick. And I'll leave that on the screen and we can jump into Q&A. We've already got a few great questions that have come in. So Jessica, first question is, how many clones can I expect to get with the CellRaft Air?
Yeah, so it's really fairly cell line dependent, but the short answer is as many as you're hoping to get. Because of how efficient the system is, and how many cells you can screen in a single consumable. For typical cell lines, I would say most customers can expect from a single array anywhere between several hundred to as many as several thousand, depending on what you're doing and how many you actually want. Realistically, most of our customers tend to isolate maybe 1 or 2 96 well plates for downstream interrogation, largely because the growth off raft is so high that you don't need to propagate 4, 5, 6, 7 96 well plates like some other technologies. So for the most part, most people are doing somewhere between 100 and 200 clones.
Okay. Do you need a fluorescent reporter or antibody stain?
Absolutely not. They can certainly be helpful to your workflow if you're looking for something specific, like a surface marker or a fluorochrome or an integration of a GFP. But because our software has been designed to be fully rooted in the bright field, you do not need a fluorochrome to interact with the technology. So you can just look for bright field characteristics like morphology or cell number or cell size without having a fluorescent marker being a requirement.
Okay. Can you find my rare event or low efficiency edit?
That's a great question. That is what the air system really excels at. Unlike limiting dilution or flow sorting, where that needle in a haystack can get lost very easily and would be very hard to find. The great thing about the air system is because we can look at all of those positions independently and recover them with a high degree of confidence, we're very, very efficient at finding rare or low efficiency edits. So in some cases, we can do that down to less than a percentage or even a half a percentage of the population. And in some cases, one of my favorite customer stories is that they were trying to do a knockout in a line that actually had 3 copies of that gene, and they were unable to derive those clones using traditional sorting methods because the triple knockouts were just too slow growing and they didn't survive. But with the air system, because they were able to select on a growth phenotype, they were able to successfully recover those triple knockouts from all 3 of the gene sites. So it's a good example that highlights how using the imaging-based technology, you can select for phenotypes that you might not see in a more bulk sorting type approach.
Okay, here's one I've seen often in our webinars. Can you use this instrument with suspension cells?
Yeah, absolutely. The workflow is slightly different and does require a little bit of optimization, but if you are amenable to adhering your cells temporarily to the rafts, it works just the same as using an adherent cell line. So we can simply coat the array with whatever coating you'd like to use, whether it's an inert coating like a poly-D-lysine or a more physiologic coating like a matrigel or a gelatin or a collagen. You can coat the rafts in order to adhere your suspension cells. And then once you've transferred them off of the array, they go right back into suspension just with simple pipetting like you normally would.
All right. How can I select the best colony from antibody producing cells?
That's an awesome question. So you've hit right on our newest application for the air system, which is finding high antibody producing cells from a bulk population.
We have developed some protocols to do that using fluorescent reporters that can look for both antigen specificity as well as just generic IgG secretion from a variety of cells. We have proof case this with primary B cells from mice and humans. We've done this with hybridomas as well as like just your producer cell lines for recombinant antibody production. It's very, very easy to do. You simply seed the cells with your detection reagent of choice, and then we can get a really lovely antibody halo around the cells. You can also do this with beads if you wish. There's a couple different ways to do it. If you are interested in learning more about that workflow specifically, we have some resources available, or you can certainly contact me directly for more information.
Okay. How long does it take from seeding to transfer into a 96 well plate?
Yeah, so in general, it's fairly rapid. Most cells stay on the air system for anywhere between 3 to 5 days, depending on the line and the growth rate. We typically want the cells to go through at least 2 to 3 doublings to get the best benefit of that shared flask environment. So most of the isolations are happening when the cells reach that 8 to 16 cell stage during division, but it could be a little longer or a little shorter, excuse me, depending on the growth rate and the phenotype that you're looking to find. End to end, most people tend to, for example, seed on a Monday and then isolate on a Friday, and you could be on and off the array in about a week. And most of that workflow is fairly hands-off, with the scans taking roughly 10 minutes and isolation running about 45. You can do that whole workflow pretty easily in about 3 to 5 days.
It looked like the CellRaft instrument was not being housed in a biosafety cabinet with a closable sash, but was located inside a hood of some type. Can you speak to how, where the instrument needs to be housed or contained for aseptic cell culture techniques?
Yeah, absolutely. So in the lab, what you are seeing is our CellRaft Air is housed in a custom hood that we can provide to you when you purchase the instrument. It is a modified laminar flow hood that's a benchtop hood, more fit for labs that don't want to use a biosafety cabinet to house the instrument because BSC space is limited. So this lets you put it in a biosafety cabinet-like environment on a benchtop like we have in my lab. It can fit in some biosafety cabinets as long as it's a 6-foot hood that is deep enough. So we do have some users who are doing, for example, more sensitive workflows like stem cells or biologics or antibiotic-free culture where they put it in a hood. And we also have plenty of users who don't enclose it at all. It's just sitting on a bench top in a clean space. You don't need to enclose it for your average workflow. We see very, very little instances of environmental contamination because of how the instrument is handled. But it is certainly possible to put it inside an enclosure if your workflow necessitates it.
Okay. What is the largest draft size you offer?
Yeah, so as we call it, the arrays come in several different flavors. They have, as you saw today in the lab, we have the smallest array format is our 100 by 100 micron square. So the cell rafts are 100 by 100 microns. The next size up is 200 by 200 microns square, and our largest raft size is 500 microns by 500 microns square, and that is our 3D array. So I didn't talk anything about that today, but we have a completely separate consumable that we can use to isolate and culture and transfer three-dimensional structures like organoids, spheroids, tumoroids, just like we do with the 2D workflow. So the 3D array is bigger, allowing us to grow three-dimensional structures up to about 1 millimeter in diameter. And again, we have tons of information on that workflow on our website if 3D workflows speak to your workflow needs.
All right, great. It looks like that's all of our questions that we have for today. So big thank you to Dr. Hartman for sharing her insights and to
Absolutely.
everyone here for joining us today and for all the great questions. Before you go, please take a moment to fill out the 2-question survey that'll pop up when the webinar ends, and we'll send out the recording link and a few extra resources in the next couple of days. Thank you, everyone.